Bismillahirrahmanirrahim. Assalamu alaikum. Shatreen Sawati is here and welcome to Biosciences with Shatreen Sawati. And today I am here with a lecture from coronavirus series, and I have delivered two lectures. First one was about the structure, and the second one is about the genome of the coronavirus, and this third lecture will be about the spike proteins of the SARS coronavirus 2. So before delivering the lecture, I have a very humble request to all of you. Kindly subscribe my channel, like my videos, share my videos. If you are sharing videos, you are sharing lectures, it means you are sharing the knowledge. So keep sharing, keep promoting knowledge. Now, I'm going to start my lecture about the spike proteins, and I want to share something with you that I created this lecture and the lectures before on the basis of the research articles. And this lecture, almost I spent 3 weeks on the creation on making of this lecture. So I have all these research articles, and if you people need research articles and any help, I will provide you. Kindly send me the email, or write in the comment box. I will share, I will help you. Now I'm going to start my lecture about the SARS coronavirus 2 spike proteins, which are very important proteins. And you can see here the very attractive arrangement of the spike protein on the surface of the coronavirus. As I have discussed in my early lecture, that these spike proteins give the coronavirus crown-like appearance. That's why this is known as coronavirus. So in this lecture, I will give the introduction to the spike proteins, then the structure, then role, and the key factor in infection, and as a potential target for drugs, and many more. Now, I will discuss all these things one by one. So what are basically the spike proteins? Spike proteins actually present on the surface of the coronavirus. As you can see here, that the spike proteins are present on the surface, and this spike protein consists of the expanded head-like structure, and the tail or stem-like structure, which is narrow. Now, there are about 74 surface spike proteins. There are almost 74 spikes are present on the surface of the coronavirus, and each spike is about 20 nanometer long and is composed of the trimers of the S proteins. This is the homotrimer S protein, and it belongs to a class 1 fusion protein, because this spike protein, S protein, help in the fusion or attachment of the coronavirus to the human cell membrane. So these are the class 1 fusion protein, and which mediate the receptor binding and maintain fusion between the virus and the host cell. Is it clear now? About 74 surface spikes are present in coronavirus 2 or COVID-19, and these spikes, the size of these spikes is almost 20 nanometer each, and they belong to the class 1 fusion protein. Now I'm moving towards the next slide, that they are the key factor in infection. These spike proteins not only present in the coronavirus series, they are also present in many other viruses like HIV viruses, because these proteins are glycoprotein in nature, and all these viruses, HIV, influenza, and Ebola virus, etc., all these have glycoproteins. The nature of the spike protein is glycoprotein. So this spike protein is present in all these viruses. Now, the role. Spike protein play a very important function. They help in the receptor recognition. Receptors on the host cell is recognized by this spike protein or S protein. cell attachment or cell fusion, and entry to the host cell. As I have mentioned that they are very important and key factor in infection. Now, the structure. The total length of SARS coronavirus 2 is about 1273 amino acids, and it is a single peptide. Must keep it in your mind. And the size of the S protein is about 180 to 200 kilodalton. And this S protein consists of 3 parts. Number 1, N-terminus. Number 2, S1 subunit. And number 3, S2 subunit. Now, N-terminus is composed of 1 to 13 amino acids. And from 14 to 685, S1 subunit. And remaining S2 subunit consists of 686 to 1273 amino acids. Is it clear now? Now, the structure subunits. As I have mentioned that each spike consists of the 2 parts, head and the tail like a stem like structure. You can say expanded portion here. This is the expanded portion. This one. This is the S1 subunit, and this is the S2 subunit. So this S1 subunit contain the RBD, means the receptor binding domain, and this S1 help in the recognition of the ACE2, means the receptor on the surface of the cells in human and other mammals, but here I am discussing about the human. So here I will discuss about what the and I will mention just human cell membrane. So this spike S1 subunit fuse with the first recognize the receptor on the host cell membrane, then attach with that receptor at the cell membrane and causes the infection. So how many units it contain? It contains 2 S1 unit and S2 unit. S1, this is the expanded portion, this is head, this is S1, and this stalk-like portion is known as S2. And this S2 help in the anchoring of the spike proteins on the surface of the coronavirus. Is it clear? Now I'm moving towards the, and you can also read these slides and note down all the material. Easily you can write and you can learn. Now, first of all, the S1 subunit. Here, S1 subunit, these are different parts of the, I have mentioned here the parts of the S1 subunit. S1 subunit consists of the CT domain. CT domain means cytoplasmic domain. Number 2, receptor binding domain, which is known as RBD domain. I have mentioned that RBD. Now, you don't confuse about what is RBD, what is CT domain. Now, in the lecture, I will use just RBD, CT, and NT. So, RBD, receptor binding domain, CT, cytoplasmic domain, and NT is the aminopeptide domain. All these domains play a very important role in fusion and attachment of the coronavirus to the host cell. Is it clear? Now, the S2. S2 domain, S2 subunit consists of number 1, TM domain, means transmembrane domain. Number 2, heptapeptide repeats, HR1. Heptapeptide repeats are present in 2 forms, HR1 and HR2. So HR1, heptapeptide repeats. HR2, and all these HR1 and TM help in the attachment, help in the fusion. So here is also CT domain is present, and most important, the fusion proteins are present. These I will discuss later on what is the nature of these FP protein, and they play a very important role in the attachment of the coronavirus with the host cell membrane. Is it clear now? Now, receptor binding domain. First of all, the receptor, what is basically the receptor binding domain is, or what is the receptor for the coronavirus on the human cell or on the host cell? So SARS coronavirus 2 S protein bind to the host cell by recognizing the receptor ACE2. And what is ACE2? A for angiotensin and C for converting, E for enzyme. This is the angiotensin converting enzyme which convert the angiotensin 1 into angiotensin 1 to 9. Now this SU2, now I'm using just the word SU2 abbreviation. SU2 is distributed mainly in the lungs, intestine. This is, you can say that that enzyme and angiotensin is that type of protein which play a very important role in immunity. So it is present in the lungs, intestine, heart and kidney and alveolar epithelium type 2 cells. These are the major expressing cells. So that's why the coronavirus attack on respiratory system, and that's why known as SARS. So SCE2 is also a known receptor for not only the SCE2 is the receptor for coronavirus 2, SARS coronavirus, it is also the receptor site for SARS coronavirus 1, which was emerged in 2003 in China. I have discussed in my previous lecture, so must watch and listen to that lecture. So the S1 subunit of the SARS coronavirus S protein bind with the ACE2 to promote the formation of the endosomes. Actually, when S1 subunit of the SARS coronavirus S protein bind with the ACE2, mean the receptor, it promotes the formation of the endosomes. And these endosomes then in turn trigger viral fusion activity under the low pH. Must keep it in your mind. I'm giving you a point, a hint that we can also stop the viral infection or coronavirus, SARS coronavirus 2 attack or the fusion with the host cell by controlling or by changing the pH. This is very important. Now, you can see here, this is the ACE2, this is the human cell membrane, host cell membrane, and this is the ACE2 receptor. This receptor is recognized by the spike protein S1 subunit, and coronavirus come and attach at that site and then causes the infection and enter into the host cell. I will discuss in detail about the life cycle of the coronavirus. So this is very important lecture, so must watch this lecture, listen carefully. And you can see here the transmembrane protease here on the host cell. And this transmembrane protease activate the spike protein, mean S1 protein, to bind with the or mediate the infection. Is it clear now? Now, how to block the ACE2 binding with the S1 subunit? Most of the researchers throughout the world working on this, that how can we block, how can we stop SU2, means the receptor of the host cell with the S1 subunit. Many work is going on. We can use different inhibitors, proteins, and messenger RNA and many other techniques and many other, you can say that enzymes and inhibitors and proteins are under observation for stopping or blocking the, to stop or block the you can say that the binding of the SU2 receptor with the spike S1 protein. Is it clear? So all over the world scientists are doing and busy in the research about all these things. And nowadays a vaccine is available which is Pfizer vaccine, and this is messenger RNA based vaccine, and I will discuss it in my next lecture, inshallah. So must watch all these lectures. I will discuss in detail about that. So now, fusion proteins. What are fusion? First of all, I have discussed the ACU2. Now I'm discussing the fusion protein. What are basically the fusion protein? Is a short segment of 15 to 20 conserved amino acids of the viral family, composed mainly of the hydrophobic residues such as glycine and alanine, and it help in the attachment or fusion of the protein coronavirus with that of what? With that of the host cell membrane. Previous research has shown that Fe play an essential role in the mediating membrane fusion by disrupting and connecting lipid bilayers of the host cell membrane. Is it clear? So fusion proteins, in simple way, you can say that these proteins help in the fusion or attachment of the coronavirus with the host cell. Now, what is RBD and HR? As I have discussed, that RBD means the receptor binding domain. This receptor binding domain present in the S1 subunit, and HR is present in the S2 subunit. So these two types of the RBD and HR play a very important role in the receptor recognition as well as with binding of the coronavirus with the host cell. Is it clear now? Now the metastable protein. S protein or spike protein is metastable. What does it mean? Now I'm going to tell you. These proteins remains inactive until they reach the host cell and attach with the ACU2, means the receptor of the host cells. They remains unstable. They remain stable until they reach to the host cell. So the spikes are also coated with the polysaccharide molecule, and this polysaccharide coating helps the coronavirus to camouflage and evading the surveilling of the host immune system during entry. Is it clear? It easily enter and camouflage themselves. These viruses camouflage themselves with the help of this polysaccharide molecule, which is present as a coat on the spike proteins. Is it clear? And must keep in mind the S protein, spike proteins are metastable. They undergo changes when they attach with the receptor on the host cell. Now I'm showing you how. You can see here the clear difference. When the spike protein S1 subunit is not attached with the, you can say that not in contact with the host cell, they are in that form. And when they attach with the host cell, they undergo the change. This is the metastability of the S protein. Is it clear now? They change when they attach with the receptor. Now, how do S protein become active? This is very important. As I have told you, the S proteins remain stable and do not change before infection and when they are not in contact with the host cell. What will happen when they in contact with the host cell? The TM transmembrane proteases, which are serine proteases, which are present on the host cell membrane, help in the cleavage of the S protein subunit S1 and S2. It play a very and activate the S proteins of the virus. These transmembrane protease on the host cell actually help in activating this coronavirus spike proteins. So the binding and fusion will be easy with the SU2 on the host cell membrane. This is the mechanism. They use the host cell proteases to become active. Is it clear? And you can read here that SARS coronavirus is cleaved into S1 and S2 subunit by the cellular proteases and the serine proteases. That's what TMSSS. So use as a protein primers for the activation of the coronavirus and for attachment and fusion and entry of the coronavirus, or you can say the infection is possible because of that mechanism. Is it clear now? Now I'm moving towards the how to identify intermediate host. I have discussed in detail about the host of the coronavirus and how it is transmitted from the bats to the we can say that from birds to bats, and then bats to birds and animals, and then from animals to human being. I have discussed. So the reason is that the SAE2, which is the receptor for the SARS coronavirus 1 and 2, is common in all these amphibians, birds, and mammals. But must keep in your mind, as you can see here on the slide, the binding affinities between SAE2 and SARS coronavirus 2, as from mammals, birds, snakes, all had found that the SU2 of the bovids are buffaloes, cows, etc., mammals, and the chrysatides are the rodents. It is indicated, it has been found that the SU2 from these are similar, but the SU2 from the snakes and turtles could not. Is it clear? That what? SU2 from different species like amphibians, birds, and mammal has a conserved primary structure. So that's why it has multiple hosts. It can affect multiple hosts at a time. Is it clear? That's why it is, you can say, circulating in all these animals and then reach to the human. And so this S protein is the potential drug target. This is very important. The fundamental role of the S protein is in viral infection indicate that it is a potential target for vaccine development, antibody blocking therapy, and small molecules inhibitors, and many more. The RBD region is a critical target. RBD region is present in which region? S1 region. Is a critical target, which is the receptor binding domain for neutralizing antibodies. And it has been confirmed that the SARS coronavirus 2 and SARS coronavirus 1 RBDs are 73 to 76% similar in sequence. Must keep it in your mind. And the 9 residues in the coronavirus RBD, must keep it in your mind that 9 residues in coronavirus RBD are fully conserved and 4 are partially conserved. So the changes occur in here. That's why the SARS coronavirus 1 and SARS coronavirus, there is a difference between the SARS coronavirus 1 and SARS coronavirus 2. They're 73 to 76% similar. About 27% differences is because of the 4 partially conserved region in the RBD region. RBD, which is the receptor binding domain, and that domain help in the attachment of the fusion of the coronavirus with the ACE2 receptor on the host cell. Is it clear now? Now, I hope so the concept is clear. Now, the notable variation in SARS coronavirus 2. As I have discussed, the RBD which is the most important target for the drug development. And as I have shown that the SARS coronavirus 2 and 1, there is a difference of about 7 to 23% differences, and 9 residues in the RBDs are conserved. Now, recent studies have indicated notable variation in SARS coronavirus 1 and 2, such as the absence of 8A proteins. And I have discussed about the 8A, 8B, 3C proteins, which are the non-structured protein in my previous lecture. So must watch that. So 8A protein, absence of 8A protein, and the fluctuation. 8A protein is totally absent in SARS coronavirus 1. And the fluctuation in the number of amino acids, and also you can see here the number of amino acids fluctuates in 8B and 3C proteins in SARS coronavirus 2. These are non-structural proteins. Non-structural proteins are those proteins which make enzymes or other proteins, but they don't take part in the structure of the coronavirus or any other viruses. Is it clear? I have discussed structure and non-structure, so must watch my first lecture about that. So the difference, number one difference, that 8A protein totally absent, and the number of amino acids fluctuate in 8B and 3C proteins of the coronavirus 2. That's why there are many differences, or you can say there are 23 to 27% differences in that. And that's why the drug used for the coronavirus 1 is not, you can say that, useful, inhibit the coronavirus 2 attack. Now, it is also reported that the spike glycoproteins of the Wuhan coronavirus is modified via a homologous recombination. Now, the reason is that the Wuhan coronavirus means the SARS coronavirus 2. The spike, which are glycoprotein in nature, modified via a by homologous recombination. The spike glycoproteins of SARS coronavirus 2 is the mixture of bad coronavirus 1 and not known beta coronavirus. There is another coronavirus which is beta coronavirus. So this beta coronavirus and the SARS coronavirus 1, this is the mixture. The mixture of both present in the SARS coronavirus 2. So many variations and mutations, and you can say that modification you can observe in the SARS coronavirus 2. Now, the major obstacle in the research progress. This is the major. S proteins are the major obstacle. Analysis of the RBM. RBM means receptor binding motif. A portion of the RBD making direct contact with the SU2. This RBM, what is RBM? RBM is the portion of the RBD, means the receptor binding domain. And receptor binding domain, what is the receptor binding domain? This is the domain which come in contact with the ACE2 receptor of the host cell. So the RBM of SARS coronavirus 2 and SARS coronavirus 1 revealed that most residues essential for ACE2 binding in the SARS coronavirus S proteins are conserved in the SARS coronavirus 2 S proteins. Mostly what? The research revealed that the proteins are both SARS coronavirus S and S2 coronavirus 1, and which is SARS coronavirus, and SARS coronavirus 2 are mostly conserved. Must keep it in your mind. In the RBD region, most of the portion is conserved. Now, however, some studies showed that murine monoclonal antibodies, must keep it in your mind, monoclonal antibodies and polyclonal antibodies against SARS RBD are unable to interact with the SARS coronavirus 2 S proteins. Means the antibodies are polyclonal antibodies, which are mono and poly, which are made for the SARS coronavirus 1 RBD domain, which are made against that coronavirus, are unable to interact with the SARS coronavirus 2 S protein, revealing difference in the antigenicity. It means the antigenicity of the SARS coronavirus 1 and SARS coronavirus 2 is different. Is it clear now? How and why? I'm giving you the question and as well as the answer of that question by this explanation. Now the concept is clear. Similarly, our SARS coronavirus RBD specific antibody failed to block infection mediated by the S proteins of the SL coronavirus. Similarly, that is also unable to block the infection by coronavirus 2, which is made for SL coronavirus. So, which suggests that the S1 RBD may not be an ideal drug target due to the highly mutable characteristics. This is very important. Must keep it in your mind. Read these points. Go through my lecture, especially from this lecture and the lecture I delivered before that lecture, which is about the structure, which is about the genome. So the concept, all these lectures will give you the concept, and you can find more and more. If you have any question, kindly share your questions, your ideas with me. I will try my best to solve your questions, and maybe you give me some points which will prove helpful for me to search out more and read more and make more lectures, create more lectures for the researchers and all the students who are taking interest in the research upon the coronavirus. So I will be back inshallah with a new lecture from my coronavirus series on the life cycle of the coronavirus. So Allah Hafiz, and thank you very much. Watch my videos, watch my lectures, like and share with others. So Allah Hafiz, thank you very much. I will be back.